Amy, you didn't remind me. You're my reminder.
Sorry.
It's okay. I've been relying on you to be my reminder in every class. Okay. So does anybody have any general questions regarding the experiment that their group is doing, the graphs that they've made, or how to do the analysis of those graphs? Okay, Amy?
Well, my question is about the record of the lab lecture. I tried to find it, but I couldn't. So it was
Not on
YouTube as well. So
I'll go back and double check because there's a processing part of it, and if my computer goes asleep or I walk away from it, sometimes it poops out on me and I don't realize that it didn't finish. So I will check on that. Okay. For last Monday you're talking about. So when I go upload this one, I'll do last Monday and this Monday. Okay?
yes, thank you.
Okay. Sure. Any other questions on data analysis, graph making? I know that Hannah's been helping, so if your group is struggling still to figure that out, reach out to Hannah. She's here today, so she can maybe put your email address again, Hannah, in the chat box so that people have it and can get to you if they need to get to you. Okay, so no other questions, are you sure? Okay, so then the next thing I want to talk about, are there any groups that still need data? Does everybody have all the data they need to tell their whole story? Okay. Just keep in mind that if you feel like, for example, I'm going to use Irwa's group, their original project was to do the zones of inhibition and to do spec reading, and I only gave them data on zones of inhibition, so they came back and they said, we really would like to have both parts of this, so I went and found data for them for the spec data too. So if you're missing something or you feel like you want more data, you got to let me know so that I can get it to you. Okay, so the next two things I want to talk about are the paper and the poster, and then I'll break us out into groups and we'll talk and then we'll be done. So that won't be a super long lab today, which it normally isn't a super long lab even if we're face-to-face. All right, so with regard to the paper, I'd like it if all of you take notes while I'm talking. And the reason I'm saying that is because this is one of those things that when you hear it, you think, oh, that totally makes sense. But then when the time actually comes, we forget what was said. So I'm going to pull up a sample paper and share it with you. That's not it. Hold on one second. I got to find it. All right. Hold on, that wasn't the right one. There it is. Okay. I'm going to pull up this sample paper and we're going to look at it together so that you can see how your formatting should work. All right. So, okay, so can everybody see the paper that I'm sharing with you?
Yes.
Okay. So you need a title page. Remember, this is worth points. Amy, are you raising your hand or just saying yes?
I'm saying yes, sorry.
Okay. That's okay. So you can see that you need a title page. This is worth 3 points. Just get it right this time and you won't have to worry about those points again. You need to have a title that's in sentence case. You can see here every word's capitalized except for the words of and in and and. And then of course you need to have your name somewhere. Make it clear that it's your paper. Don't just list your name and then the whole group's names together so I don't know whose is whose. So put your name here and then put lab group somewhere else down below, okay? That's worth 3 points, right, of a 100-point paper. So if you forget the title page, and I still have people turning them in without it, like the highest grade you could possibly get to 97. So don't do that, okay? All right, now when it comes to the abstract, and we didn't talk about the abstract too much last time, so I want to make sure we take a few minutes and talk about it now. The abstract goes at the beginning of the paper. It's the very first thing after the title page. It's before the introduction. When you were looking at papers online, you were looking at abstracts. That was basically what you were reading to see what the paper was about. So your abstract should have minimal 3 sentences. It could be 5, okay? The first sentence needs to be the big picture. So if you look here, it says, in today's world, there's a growing concern about the increasing level of antibiotic resistance among the animal population. All right, so that's what this is going to be about, right? And then somehow tie what you're doing into that big picture. So research continues to seek to explain how our health is affected by different foods. This study was performed to determine whether not eating organic versus non-organic food might have an effect on the occurrence of antibiotic resistance of bacteria and consequently cause potential infections that are difficult to treat in humans. Okay, so now I know, okay, it's a big picture problem about antibiotic resistance. They're looking at diet to see how that might influence that. And then, so that was your second sentence. First sentence is big picture. Second sentence is somehow relating your project to the big picture. And then a brief statement on how you're going to do this. In order to determine this level of resistance, organic and non-organic food broths were cultivated with this bacteria. Antibiotics were introduced and zones of inhibition were measured. So I'm not telling any specific details. I'm just kind of giving you the general gist of here's kind of what we did. All right, so that was sentence number 3. And then the fourth sentence would be a summary of your results. It was found that S. epidermidis thrived best in non-organic food broth, but data regarding the level of antibiotic resistance were inconclusive. It's okay if you have inconclusive data. This is your first ever doing an experiment. People do experiments for years before they get something super conclusive. So you don't have to have, oh my gosh, I found something novel and I can publish a paper. That's okay. It can totally be the results of this study did not show what or support our hypothesis. The results were inconclusive. The data was unclear. The cultures were contaminated and no data was received. Whatever it was, that's what you have to state. Okay? So, abstract, I would write it at the end of your whole paper. So, even though it's the first part of your paper, I would put off writing it till you're done and you've written your whole rest of your paper. Any questions on the abstract before we move forward? Notice that there is the word abstract above it. Oops, let me see. Right, the word abstract is right here. So you need that subheading, and then you have your paragraph that's your abstract. Can you hear my cats meowing?
Yeah, they sound like children.
Yeah, no, it's just, I don't know why she's meowing, but she's meowing like crazy. I don't know if she's like stuck somewhere or what. Okay, any questions on the abstract?
No.
We're good? All right, the next, so that's something new that we haven't written yet. So that's going to add, and I can't remember off the top of my head how many points that's worth, but let's pull this up. I can look. It is worth 6 points. All right. So if you don't use that appropriately, then you're going to miss out on those 6 points. Okay. Next, we're going to go to introduction. Can you see the introduction here?
Yes.
All right. So remember, your introduction has to have a paragraph comparing prokaryotes and eukaryotes. Right. So this paragraph does it. You have to have some citations. This isn't quite APA because it should just have this last name, comma, this date. You don't need all the other things. All right. Then you have to have a paragraph on the domain that you're choosing. So in this case, they're talking about domain eukarya. And then you have to have a paragraph on the organism that you're studying that fits into that domain somewhere. And so tell me where it fits in. And then tell me a little bit about that organism. And as much as you can during that part, you want to relate somehow tell me information that's relevant to the big picture question. So for example, this person says staff are pathogenic. And then they tell me a little bit about them. The colonies are round and gray. This is usually harmless, can be found on the skin and in your nostrils. However, if you get a puncture wound, it becomes a problem and can cause infections, especially in hospitals. And so I'm seeing where they're going with this. If these are causing infections, then antibiotics would be the typical way to treat them. All right. So then you want to relate that back to the big picture question. So this person has a really long. So this purpose of this experiment is to determine if there are any significant differences between the levels of antibiotic resistance when grown in these different things. All right. So I think most of you have a pretty well done introduction. You might not have that little paragraph relating your particular organism, your study, to the big picture. So make sure you put that at the end of your introduction. And that is worth, can you see this? Did I just put this up, or no, you can't see this? It's
No.
the rubric. You still see the paper. Okay.
Yeah.
So on the rubric, which I'll share with you in just 1 minute, that introduction is worth 24 points. So 12 points based on what you've already turned into me, and then an additional 12 points to make sure you have that big picture related to the project at hand, and make sure you kind of have stated what your expected results are. All right, so that's worth 24 points. All right, the next section is the materials and methods. And you can see here, they have a little paragraph at the beginning kind of restating the whole purpose of this experiment. I think it's a good idea to do that because sometimes people only read one section of your paper, and so this tells them, okay, this is what you're doing, but they did a little bit, maybe too much that they needed to do. All right, then you're going to have whatever method you use, and you need to have two methods. You need to have the characterization of your organism method, which is what they're calling gram stain, and it has to have a subheading, so you can do whatever you want. Characterization of, oh, you still see the introduction paragraph? Okay, hold on. Let me see. It says I'm sharing it with you. Interesting. All right, I'm going to stop sharing and reshare. I don't know why that happened. I guess maybe because I clicked away. Okay, now can you see methods? You have to tell me.
Yeah,
Yes.
it's working now.
Yes.
Okay, so so in your methods, can you see it now?
Yes.
Let me see something real quick. I feel like now I'm not on the same page you guys are on anymore. Okay, so in your methods, you're going to have the word methods bolded as a subheading, and then under that, you're going to have two subheadings. One is about the characterization of your organism, and you're not going to say organism, you're going to state what your organism is. So you can say characterization of E. coli, or morphology and size of staph epidermidis, or something like that, okay? Has to be informative. And you're going to have a very quick couple sentences on that. To confirm the thick-walled nature of these cells, that's kind of a weird way to say it. This was actually a student's paper. A gram stain was performed. Then it was viewed under a calibrated microscope to observe the physical properties and measure the length of the cells. So remember, we're talking under this subheading, you're talking about visualizing it under the microscope. If you did any sort of staining, that has to be included. And then you have to talk about measuring. Okay? And then you're going to have a separate subheading that talks about whatever your experiment was. So the effects of essential oils on the growth of your organism, the effects of nitrogen on the effects of your organism. So that should be this subheading right here. Okay? and then you're going to talk about that. And you guys already wrote those and you turned them into me, and it's okay if you share among your group. That's totally okay with me. Hey, Keith, don't talk in here. Okay, any questions on method? All right, now this part's new. We haven't done this yet. So the next section is called results. And this one, again, there's a little paragraph here kind of restating what this study addresses. I think that's a good idea because sometimes people only read the results section. So if you don't have that, then they're not in sync with what you're doing. And then you have to have a result for each method you listed. So for groups that have, if I go back up here, for the groups, for example, Irwa's group, where they did 2 different experiments to test the growth of their organism. They did the zones of inhibition and they did the spectrophotometer readings. She might want to have 2 subheadings: effects of, I can't remember what disinfectants, was that what it was, or what? The effects of different disinfectants on E. coli using zones of inhibition, and then she'll have a separate one that says the effects of different disinfectants using a spectrophotometer, and then she'll list both of those results separately in the results section. Okay, now this is what I want you to write down. This is really important. In your results section, only report results, no interpretation of the data. So you don't have to say this means it has a thick cell wall or this means that it's a prokaryote. All you're going to do is state what you found. All right. So in your results section, you're going to say the cells were gram stained and visualized under the microscope. The cells were round and they stained purple and they measured to be this size and diameter. That's it. See figure 1, and then you're going to have a figure 1 that has a caption that basically restates that same information. Then you're going to go to your experiment. So again, you might say, to understand how this works, this is what we did. This is what we found, that, with Clorox, the cells were killed. When we use just plain water, the cells weren't. And you're actually going to say the numbers. So you're going to state the millimeters of your zone of inhibition, your absorbance for the spec readings, or I guess those are the only 2 things we're doing. If you're actually measuring cell size, you would say the diameter of the cell. Okay? And then you're going to refer to every result to the picture. So you may not have pictures like this because you guys didn't actually do the experiment, but you'll refer to your graphs. All right, so here they have their graph of the effects of the antibiotics on this organism, and you can see in the organic one, the size of the cells were this, and the non-organic, they grew like this, and so that showed that there was a difference in this particular group. Only state results. Okay? Your raw sets of data, which are the tables that I provided to you, those are going to go at the end of your paper in the appendix. All right? So you don't put those in the results section. All right? I'm sorry about the people talking. You guys, don't talk. Okay, and then the discussion is the last part of the paper. And this is where you get a little freedom to interpret your results. So you're going to kind of tell the story again. You're going to restate another time. It's going to seem very repetitive, but this is the way science papers work. You're going to restate the purpose of the study. You're going to restate what you found in your results, and then you're going to interpret what those results mean. For every result you find, you're going to interpret it. Now, that doesn't mean for every little data point, but you can summarize, here are the results. The essential oils of lavender and oregano oil effectively reduce the growth of E. coli, give us some data points, and then tell me what it means. All right. Also in the discussion, you're going to include maybe some places where maybe the information could have been collected better. Maybe you would have rather asked a different question. So you can talk about mistakes you may have made, which you guys didn't really make any mistakes because you didn't do it, or future directions. Like, given this information, now I wish I would have asked the question this way and what I would do in the future. All right. You need to be careful during the discussion to relate your findings to the big picture, to your original hypothesis, and to the literature that's out there. So in your discussion, you should have some citations. This study, the results of this study support what Joe Schmo found, in 2018, or maybe it didn't support, but just mention, like, the papers that you read. Does your data go along with those papers, and refer back to them? And then the last part of your paper is your works cited page that has to be in APA format. It looks something like this. This one's done right. All right. And then the last, after that, then you'll have your appendix with your tables of your data. And you all have those tables this time because I provided everybody with those tables. All right. Now this paper is due this coming Saturday by midnight. All right, so there's a Turnitin link on Blackboard, and so it'll be due this coming Saturday by midnight. Any questions about any of that stuff? Is that clear? Amy, yes, clear?
Yeah, so this one is going to be our final final paper?
Well, it's kind of weird the way we word it, but yes, but you still get one more chance to do edits. So it's not really the final version you can turn in. Our hope is that this is as final as it could be, and then I just send it back and I say, okay, you missed 10 points on your results section. You have one chance to still make this better. Okay, so let me stop sharing this with you and answer Grace's question. So is there a rubric? This is in the Blackboard, the Google Drive that we share with you all that gives you the rubric. So here's the rubric. Here's how it's graded. And so Karen just said, do we get a grade from this one? Yes, this is the grade that goes into the grade book this time. However, if you send me a revision and there's a change to the grade, I'll change it. So let's just say you get an 80 and I say your results section really needs some work still. So you get a 9 out of 28 on the results section. And I say you have an opportunity to rewrite that part. If you rewrite that part and you earn 10 more points, I'll go in and change your grade from an 80 to a 90 in the grade book. So yes, what you get on this one will be a grade in the grade book, because if you choose not to do the revisions, which you totally can not choose to do, then that will be your final grade. Keep in mind that this is worth 40% of your total lab grade. So it's a huge, this is one of those times where this has a huge impact on your grade. I always say, like, don't worry about pre-lab quizzes and in-class quizzes and pre-lecture quizzes, because each of those is such a minimal contribution to your grade that don't worry about it if you don't do well on one. This is one of those times where I'm not saying that. This, worry about, because if you don't do well on this, it has a huge impact on your overall grade in the course. So you want to do as good as you possibly can. And this rubric is there for you, so you can go through and ask yourself, did I do these things? Did I provide information about how I looked at the cells under the microscope and how I measured them? Did I describe why each procedure was used? Did I not have bullet lists? Did I not provide any unnecessary information? For example, don't interpret your data or even state results in the methods. That would be unnecessary information, right? So you can ask yourself all those questions as you go along. All right, any questions on that part? on the paper?
No.
You can always email me if you have any as well. All right, that's due Saturday by midnight. The other thing you have to work on for this, let me see if I can find it on my screen, for this upcoming week is your poster. All right, now you have 2 weeks to work on this, so you don't feel like you have to have this all done. And really the poster is just a reflection of the paper, although you get to do it as a group. So let's just look at the poster template. So you can kind of start. One of the things you might want to start doing this week
Hang in there for a second. It looks like she disconnected. Amy, are you there?
Yes.
Yeah, I had a question for you about your graph. So I'll ask you about it before you leave, okay?
Yeah, sure. I'll stay. Thank you.
Sorry guys,
cool.
I'm back, and I don't know what happened. I got kicked out. Can y'all hear me?
Yeah, we can hear you.
So sorry about that. So so the next thing you need to be thinking about is the poster. So can you see this poster template that I'm sharing with you?
No.
No, I can't see it. Let's see. Let's see. Why are you not seeing it? This is what crashed me last time, so hopefully it didn't crash. Doesn't crash. Can you see it now?
Yes.
Okay. So, oops, I just shared the wrong one with you. Hold on one second. I don't want that one. I want. Okay, now you can see it. All right, so this is kind of what your poster is going to look like. There's a template for this in PowerPoint, in Blackboard, in the Google Drive. I'm sorry, not in Blackboard, in the Google Drive. So all you have to do is really just put your poster together like this. So what I think you could work on this week is divvying up the duties. Who wants to be in charge of, maybe this column of the poster? Most of my groups are groups of 3. So who wants to do this? Who wants to do this? Who wants to do this part? And really what this comes down to is taking information from your paper and condensing it into a form that fits onto the poster. The abstract, you can just find out who in your group wrote the best abstract and then put theirs here. All right. For your intro, it's not going to be your whole entire intro. It's going to be more just the big picture and here's what I did. The methods can kind of be what you wrote in your paper. That little paragraph you all sent to me is fine. And then in your results section in this middle, you'll have a results of your characterization of your organism. So you're going to have a picture of your organism. I think everybody has one now with a caption. And then you're going to have a graph with a caption for your experiment. Okay? And then on this side, you'll have a discussion. And it doesn't have to be your whole discussion. It can kind of just be the highlights. This study supported the fact that, antibiotic resistance is a problem, and here's what the results mean. And then you have your literature cited. And then, of course, you should have an acknowledgments where you probably want to thank Trey. Hannah, you want to put in the comment section their names, but Trey and Hannah, first name and last name, because those are people that have really probably helped you. And if you want to thank, for example, Alexis, I think it was you that was asking me, what do I do if Bree dropped, right? You can put Bree in the authors, but if you don't want to, you can put Bree's name down in the acknowledgments for helping. If you have a member that was there and then left, you can do that. This is not due in a final draft next week. So next week during class, what we're going to do is we're going to take time as a group to work on this together. All right, so this is just something you want to maybe start thinking about. If you could divvy it up into who's doing what, then next week you can be responsible for bringing that condensed version of the intro or condensed version of the discussion and start to build this and put it together as a group next week. And then the following week, you'll present it to the rest of us. So what I'll do is we'll just pull up yours and you can talk and tell us what your findings were. Okay. Any questions on the poster? Amy?
Yeah, so right now the paper that we're gonna turn in this Saturday that we gonna work on our all, and after that we will share the data to work with the other member.
Yes. So you'll divvy it up into however many people there are. Everybody will take their percent, right, and do part of the poster. Yes.
Okay. Thank
And
you.
then I would say that whoever, when you're presenting your poster, whoever does the side with the intro and the methods will say, here's the big picture, here's why we're doing this, here's what we did, and then who was ever in charge of the results says, here's what we found, and then who was ever in charge of the discussion says, here's what this finding means. And so you're responsible for speaking about the part that you were in charge of. Yeah? That makes sense?
Yes, thank you.
Okay. So I am done with what I needed to say for today. It's up to you. I don't know whether the breakout groups here help because Hannah and I can pop in, or if you already are meeting as a group offline, you want me to do the breakout groups here to talk about your graphs, or to just talk as a group and figure out who wants to do what? Nobody saying anything? How about
Are we discuss about the poster, or you mean the paper that we have to turn in this Saturday?
the poster, I would say. Let's just do it. I'm just going to put you in your group. So Alexis, you are with Karen, is that right? Okay, so Alexis and Karen have redropped, so it's just the two of you. Ashley, you are with Ivan and Amy, is that right? All right, so now you guys can talk. Elaine, you are with Gretchen and Grace. All right. Jessica, I don't see that Reggie's here, so hold on for you for a second. And then Gemini, you are with Camelia and Irwa, right? And then Mohammed, who's in your group? Mohammed is with, I can't think now.
And Ramon, they're not here.
Oh, so Ramon and, okay. So let me do these breakout groups and then I'll talk to Jessica Mohammed in the big group, okay? So hold on one second. So go ahead, you're gonna get to your groups, just start talking and Hannah and I will jump in. Okay, so Jessica and Mohammed, you guys can log off if you want, but before you do, I want to make sure you don't have any questions about what to do with the poster. So you'll have to contact your partners and say, hey, we have to divvy this up, and so what part do you want? Or you can even, since you were here, you can say, hey, I want to do this part. So the other two of you have to figure out what part you want to do. Okay, do you guys have any questions on how that part works?
No, not really.
Okay, and do you have any questions about your graphs or your data so far? Jessica, I think I've seen some of yours, but I'm not sure I've seen Mohammed, your groups yet. Did you guys turn in?
I'm not sure how are we supposed to turn it in. I did a Excel and I did submit online. I don't know if you can see it or not. I just want to make sure you got it. Also,
how did you turn it in to me? Did you email it to me or did you post it in Blackboard?
No, I just did, but I just submitted it on Blackboard.
Okay, I haven't checked there yet, so it's probably there. I'll check. If I can't find it, I'll email you.
Sure. Other thing, the graphing assignment, the one on the paper, I think it was number 2. I did send it to you by email. I just took pictures from my phone and sent it by email. I don't know if you can see them well or you need me to scan them as a PDF. I didn't turn it in online, just by email.
Okay, I will look for it. And I'll let you know if I wrote it down, so now I'll let you know. If you don't see a grade come in the grade book, you might want to email me and say, hey, don't forget that's in an email, just in case.
Sure.
Okay, do either of you have any other questions? Okay, you're free to hang out if you want, but you can also log off if you want. I'm not really making any other announcements, just that tomorrow's lecture is at 12:30. I'm posting the voiceover PowerPoints today, so if you like to watch the PowerPoints before lecture, sometimes that helps. So those will be on YouTube today. I'll post this session on YouTube, and and I've graded all the tests, and your grades are in the grade book. So if you have questions, let me know.
Are you done with the office hours for today?
Yes, you have. So do you have questions? Ask me now.
Yeah, I would like to talk to you in private. If
Oh,
that's
okay. That's fine. So maybe just hang out, just go do something else, but stay logged in, and at the very end, after everybody logs out, we'll talk.
Okay, sounds good.
Can you do that? Okay. All right. So I'm going to go to a different group. So Muhammad, I'll see you at the end. Jessica, it's up to you whether you stay or you go.
Okay.
All right,
Okay.
thank you.
Mhm.
Mohammed, are you there?
Yeah.
You here all by yourself?
Yeah, I'm just waiting for Mrs. Berg.
Okay.
Yeah, thank you.
Yeah, of course.
It's the journal article research paper. It's on page 23, page 23.
You put right what you looking at the data that you have, what you put right now is the mean, so you don't have to put like
Amy, you're in the big group now, so hold up one second, okay? I'll put
right.
you back in the group in just a second. It's okay. All right, guys. So some of my groups are done, so I want to make sure that they can log off and hear the rest of the announcements. So here's what's to come. Tomorrow we have lecture at 12:30. I will post the pre-recorded PowerPoints today into YouTube for chapter 13 and 17. I'm hoping to cover both of those chapters tomorrow. So look for those this afternoon. Log in tomorrow. Your paper is due by midnight on Saturday. That's the only assignment you have for lab this week. No other graphing or something like that. And then start to divvy the duties for your poster. All right. And bring that information with you next week for lab. Any questions on what's to come? Okay. If not, I'm going to put Amy and Ivan and Alexis, or no, is it Ashley? It's Ashley back into your group, and I'm going to put Gemini, Camellia, and Irwa back in their group. If you're done, like Elaine and Grace and Gretchen, if you're done with me, you can go ahead and log off. Okay, so hold on one second. I'll do that. All right, so let's see. Two, and two, and Mohammed, I'm going to put you in your own group, and then I'll come and talk to you in that group, okay?
All right.
It'll just be a private group. Okay. Hannah, I'm going to talk to Mohammed privately, so you can go in the other two groups, okay?
Yeah, of course.
Okay. Irwa, are you here? Oh no, she's back in there.
Can we leave?
Yeah, you can leave.